In this video, I am going to explain about plasmodium vivax in man face. So a lot of information I am provided for you. So watch this video till end. You clearly understand about the plasmodium vivax in man face. So generally, plasmodium vivax will occur in two phases, in man phase and mosquito mosquito phase. So basically plasmodium vivax is a parasite. Plasmodium vivax is a parasite which causes diseases called as malaria. Okay, so now listen properly. So basically plasmodium undergoes the process of asexual reproduction, a sexual reproduction called zygogony. Zygogony. So when they enter into the liver cells, uh, this zygogony infect the liver cells and RBC. Once they enter into the liver is nothing but hepatic. Liver cells means nothing but hepatic hepatic cells. These hepatic cells the process asexual reproduction process is going on in this man phase, so it is zygogony. So it is occur in the liver cells called hepatic zygogony. In red blood cells, erythrocytes, red blood cells is nothing but erythrocyte. So erythrocytic zygogony. In plasmodium infected mosquito which consists of plasmodium and go and bite the human being, then it starts releasing 2000 sporozoites. They are the involved in the several stages. They are involved in several stages in Plasmodium vivax in man. Plasmodium vivax in man. What are they means? Pre-erythrocytic cycle, exoerythrocytic cycle, sorry, pre-erythrocytic cycle, exoerythrocytic cycle, pre-patent period, erythrocytic cycle, incubation period, and formation of gametocytes. Gametocytes. See, you have to listen properly. I am going to explain life cycle of Plasmodium vivax in man face. Plasmodium vivax in man face. Man face. Plasmodium infected mosquito. Plasmodium infected mosquito will bite. Plasmodium infected mosquito will bite. Plasmodium infected mosquito will bite human being. 2000 sporozoites will enter. 2000 sporozoites will released out, and that released sporozoites will enter into the hepatic cells. This is hepatic cells released into hepatic cells. Then what happen? It will get transferred into the trophozoite. It will get transferred into the trophozoite, and trophozoite will consumes that content. Trophozoite will consumes that content mainly present in the hepatic cells, and it develops into the zygont. This zygont is spherical in shape. This zygont is developed in sizely and increased. Zygont will undergo the process of mitotic divisions. Zygont will undergo the process of mitotic divisions. Mm, and such that way nucleus and cytoplasm uh, are grown develop are grown are developed increasingly severally. rally and such a way that cryptozoites and such that way cryptozoites are cryptozoites are formed. Cryptozoites are formed. Uh, Cryptozoites will released to 1200 cryptozoites are released. These cryptozoites are also called as first generation merozoites. First generation merozoites. This process is known as pre-erythrocytic cycle. This process is known as pre-erythrocytic cycle. This pre-erythrocytic cycle duration is 8 days. Duration 8 days. 8 days. See, in cryptozoites will have two options. In cryptozoites will have two options. One is enters into the newly fresh hepatic cells, enters into the fresh hepatic cells, and another one directly enters into the rbcs, erythrocytes. rbc, erythro red blood cells, erythrocytes. See, this exoerythrocytic cycle process will same as pre-erythrocytic cycles. Same as pre-erythrocytic cycles will get uh, cryptocytes will get transformed to the trophozoite. This trophozoite will developed and mature the vacuole, developed and mature the zygont and meta macro metacryptozoites and micro metacryptozoites will develop. Macro metacryptozoites and micro metacryptozoites are will developed. These mm, macro metacryptozoites are re-entered into the hepatic cells. Fresh hepatic cells again. Hepatic cells again. These microcryptozoites are enters into the blood or blood cells, RBC, red blood cells. This process is known as exoerythrocytic cycle. See, now see, these micrometacryptozoites will uh, enters into the RBC and will get transformed to the trophozoites. Will get transformed to the trophozoites. Will get transformed to the trophozoite. This trophozoite, this trophozoite will develop. This trophozoite will develop, mature, and the vacuole inside the vacuole inside, and the vacuole will be push peripheral region. Will be push peripheral region. It leads to the towards the nucleus వైపు వెళ్ళిపోతుంది. అప్పుడు ఎట్లా కనిపిస్తుంది అంటే ఇట్ లుక్స్ లైక్ సిగ్నెట్ రింగ్ స్టేజ్ లాగా కనిపిస్తుంది. సిగ్నెట్ రింగ్ స్టేజ్. దిస్ vacuole vacuole will be reappeared in the stage of amoeboid stage. This signet ring stage transform to the amoeboid stage. In this pseudopodia will develop. Pseudopodia will develop for movement కోసం pseudopodia develop అవుతుంది. In this RBC pigment as this is RBC pigment. This is what is RBC pigment? Hemoglobin is RBC pigment. And with the pseudopodia consumes red blood cells. Pseudopodia consumes red blood cells consume అయి ఉంటాయి. So now consuming hemoglobin part, it will develop size and it developed and pseudopodia will develop and ఫామ్ ది సైజాంట్గా ఫామ్ అవుతుంది. బిఫోర్ బిఫోర్ that hemoglobin are to heme heme and heme and globin. Heme is insoluble crystalline. Heme is insoluble crystalline, and globin is soluble crystalline. Heme is insoluble crystalline. That nothing but hemojoin. Hemojoin. It is a malaria pigment. Nothing but hemojoin. It is a malaria pigment. Malaria pigment. After this amoeboid stage, the zygont will forms. Inside the zygont, the Schaffner granules will Schaffner's granules will Schaffner's granule will developed. After this process, the merozytes will develop. Uh, when the merozyte when the merozytes when after merozytes will develop, when the merozytes ruptures, uh, it released it released out. When merozytes released out, i- from these merozytes, the hemogen also present. This is malaria pigment. This is a malaria hemogen is a malaria pigment. These merozytes have two types: female gametocytes and male gametocytes. Female gametocytes and male gametocytes. Larger female gametocytes is also called larger macrogametocytes. Male gametocytes is also called smaller microgametocytes. Smaller microgametocytes. Gametocytes. Smaller microgametocytes. Hmm. This female is larger than the uh, male gametocyte. Female is larger than the male gametocytes. This re-entered into the RBC. When the merozytes are re-entered into the RBC, this is the uh, complete of erythrocytic cycle. This is the complete of erythrocytic cycle. This is the complete of erythrocytic cycle. This is erythrocytic cycle duration is 48 hours. 48 hours. This erythrocytic cycle is also called Golgi cycle. Golgi cycle, because this erythrocytic cycle discovered by Camillo Golgi scientist. Camillo Golgi scientist. So it is called Golgi cycle. Golgi cycle. uh, Plasmodium vivax in man phase will discovered by the Garnham. Sortan Garnham. See here the stages. See the stages here. Pre-erythrocytic cycle we explained. I explained you. Pre-erythrocytic cycle I explained you here. Uh, Pre-erythrocytic cycle. Exoerythrocytic cycle also I explained you. Pre-patent period. I didn't. Erythrocytic cycle. I explained you. What is pre-patent period? What is incubation period? And formation of gametocytes. The interval between the first entry of plasmodium into the blood in the form of sporozoites. The interval between first entry of plasmodium into the blood in the form of sporozoites, and the second entry of the plasmodium into the blood in the form of cryptozoites is called pre-patent period. pre-patent period. Incubation period. What is incubation period? Incubation period. What is incubation? The period between entry of plasmodium in the blood in the form of sporozoites. The first appearance symptoms of malaria in man is called incubation period. The period between entry of plasmodium into blood. The period between entry of plasmodium into blood, entry of plasmodium into blood, and in the form of sporozoites ద్వారా బ్లడ్లో ఎంటర్ అయినప్పుడు ద ఫస్ట్ అపియరెన్స్ ఫస్ట్ ఏమైతే ఫస్ట్ అపియరెన్స్ సిమ్టమ్స్ అపియర్ అవుతాయో మ్యాన్లో మలేరియా సిమ్టమ్స్, దాన్ని ఇంక్యుబేషన్ పీరియడ్ అంటారు. ఇంక్యుబేషన్ పీరియడ్ అంటారు. Formation of gametocytes. What is formation of gametocytes? After repeated cycles of erythrocytic zygogony, when formation of gametocytes after when the number of fresh RBC decreases and some melocytes enter into the RBC and transformed into these gametocytes instead a continuing erythrocytic cycle. This process is known as uh, erythrocytic. This process is known as wait. This process generally takes place when the RBCs are present in the spleen and bone marrow. These gametocytes are two types of microgametocytes and macro gametocytes. Microgametocytes are male, macrogametocytes are female. These gametocytes cannot undergo further development in the main temperature. Man has the temperature and the pH of the blood man not suitable for the further development. These gametocytes reach the blood circulation and wait to reach the next host. That degenerate and die if they are not transformed to mosquito within a week. ఇప్పుడు మస్కిటో బైట్ చేయలేదనుకో 1 వీక్కి, ఈ ఇవేమవుతాయంటే గ్యామెటోసైట్స్ ఆర్ డీజనరేటెడ్. Okay, thank you.